Welcome to Science Philosophy in a Flash, a mini podcast series produced by the Scientist Creative Services team. In this series, we highlight researchers' unique outlooks on what it means to be a scientist. David Liu is a professor in the Department of Chemistry and Chemical Biology at Harvard University. His philosophy of science involves an appreciation of the fundamental principles in chemistry and protein evolution, which guides his research on CRISPR-based editing techniques. In their latest work, his research team took a no-stone unturned approach to determine a one-time base editing strategy for treating the motor neuron disease spinal muscular atrophy, or SMA.
As a chemistry undergraduate, I fell in love with the idea of manipulating molecules at the individual atom scale to tailor those molecules to have desirable properties. I did my PhD in the lab of Peter Schultz, and my PhD project was to create a system that would allow genetic code expansion in cells. And it became quickly apparent during that PhD project that we would need to do a fair amount of protein engineering. So I became really interested in evolution. When I started as a professor in 1999, I already had a deep reverence of both chemistry and evolution. And that's really the intellectual foundation of our lab. SMA is the consequence of these two genetic misfortunes. A gene called SMN1 was duplicated imperfectly into a closely related gene called SMN2 that differs only in a single mutation. And the result of that is that the SMN2 copy generates a protein that has a very short lifetime and is unable to do its job to a full extent. So that's mistake number one. We all have that mistake. The second mistake occurs in SMA patients, and that is that they've lost both copies of working SMN1, the one that they inherited from mom, the one they inherited from dad. Neither of them work normally anymore. Because SMN2 differs from SMN1 by a single point mutation, we became very interested in the base editing of SMN2 into a healthy copy of SMN1. Base editing and prime editing use the programmability of modern gene editing agents, such as the CRISPR-Cas9 nuclease, to find the target DNA site. But instead of cutting that target, they perform chemistry directly on DNA to convert the starting sequence to a sequence that you specify. This gene editing strategy should be applicable in theory to all SMA patients, regardless of how they lost their SMN1. Virtually everything we do is an integration of chemistry and evolution with the goal of developing new therapeutics and studying biology.
Thank you for listening to Science Philosophy in a Flash. To learn more about Liu's research, please check out the link in the episode notes.